Hey there, welcome back. We're going to continue on with the heart now for Anatomy and Physiology 232. We kind of went over heart anatomy in the last video, and now we're going to be looking at heart physiology. We'll see how far we get today, whether we break this into a couple different videos or just do one long one. We'll see how it goes. So let's switch over to our lecture notes. We left off here on bottom of page 7, heart physiology. This first part is reminding us a lot about cardiac muscle cells and what cardiac muscle cells look like. And you have a picture here, not a very good picture, but a picture here nonetheless of cardiac muscle cells. One thing that we want to remember is about them, and what's mostly underlined here, is that they are similar to skeletal muscle in that they have striations. They use actin and myosin, sliding filament mechanism, to contract and shorten. However, they're shorter, they're more branched, and they're interconnected. And they're interconnected with intercalated discs. And intercalated discs have gap junctions and desmosomes. And gap junctions, remember, help with increasing the speed of conduction because they allow ions to go through from one cell to another. And this allows the heart muscle to act as one, as a single unit, to beat together. So once a contraction starts, it will continue in a wave-like fashion across the muscle. So we like that. And the intercalated discs are listed here, or kind of shown here, with these little lines between the different muscle cells. And I wrote intercalated discs there, although it's a little hard to see. The other thing is there's very large mitochondria. There's more ATP available that's needed because your heart is working 24 hours a day. So that's important. And there are a couple of like structural differences. The T-tubules are a little different, things like that. We're not going to focus on that too much. We're going to talk about some other physiological things that are different about the heart, but right now that I'm not too worried about. Now, one thing though, as I just said, is we need an ongoing supply of oxygen, and this is because it has to be in aerobic respiration all the time. You can't go to anaerobic. We'd get oxygen debt, the muscle would stop working as efficiently, and you'd have problems. One way that the cardiac muscle can solve this is, in addition to using glucose, it uses fatty acids a lot more effectively than other body cells to make ATP. And so that's a good thing, is it can use both fatty acids and glucose to get as much ATP as possible. Now, as I was saying earlier, the way that the cell contracts is really similar to skeletal, and we're not going to go into too much detail about that because the same basic things happen. There's T-tubules, there are sarcoplasmic reticulum, there's calcium that leaves the sarcoplasmic reticulum. It allows the actin and myosin to attach together. ATP causes recocking of heads and shortening. One thing, though, that I want to mention is that calcium has another role that we don't often see. And actually, there's a couple ions that have other roles here. And this picture, you'll see my notes say it's on page 689 as well. And I think I have it up here right now, if I remember. Oop, maybe not there. There we go. That was for another class. Right here, figure 1815. On page 689, and they show that pretty much the same thing. However, their plateau kind of slopes down, which is more correct. In our picture, which is a little less correct, but maybe a little easier to follow, the plateau stays directly flat. But I'm going to use this picture because I think it's pretty good, and I'll try to explain the difference here. So between what we see in a normal action potential and contraction in this one. Okay, so first thing is what you're looking at is you're looking at it really an action potential graph, right? Think back to 231 when we were looking at generating an action potential across a membrane in a neuron, and we do that same thing in a skeletal muscle, sarcolemma, and we do the same thing in the sarcolemma of cardiac muscle. So at first things start kind of normal. You can see here, I'm going to just use my pencil to kind of highlight things. We go up, here's negative 70, which is threshold right there, right? There's our threshold. And if we reach threshold, we depolarize. So we get a graded potential, and then we get a depolarization. Now, if you remember the original action potential, it kind of went up like this, came straight down, and went like that a little bit. And here, let's redraw that. Went up like this, came back down, and then kind of went like that. And threshold was right about there. And now, so that's what it looks like. Now notice what's different here. We have this immediate drop where 1 is, and then we have this plateau. We don't see either of those. We don't see quite the sharp drop, and we don't see a plateau. So what's going on there? Well, the first immediate drop is normally what happens during depolarization right here is we have sodium rushes into the cell and potassium will go out shortly thereafter. With that sodium rushing in makes this very positive, right? Now, in the case of heart muscle, chloride immediately starts to go in after depolarization. So even though sodium rushed in and this made this very positive, a negative ion follows closely behind, and that starts to bring things down a little bit here. Okay, so that initial drop is due to the chloride ion following the sodium shortly after depolarization. Now, the next thing that happens is normally we would just start to drop back down. That's not what's going to happen because calcium is going to rush in, and as calcium rushes in, normally we start to have depolarization. The calcium's rushing in, which is making it stay positive. So those are canceling each other out and are staying relatively flat, or if we saw in the book, it starts to drop but very slowly. We call this our plateau phase. Now, where's that calcium coming from? Well, interesting, free calcium is actually outside of the cell. I'll underline this so you can see this here. Free calcium is outside of the cell, and it rushes in and it activates the other 80% of calcium in the sarcoplasmic reticulum. So we've got a little bit of calcium outside that rushes in during a depolarization and activating the rest of the calcium. So a lot of calcium comes in during contraction. This plateaus and goes and kind of extends out the action potential. Why? We'll get to that in a second. But then, as we start to repolarize, the calcium goes back into the sarcoplasmic reticulum and goes back outside the cell. The potassium continues to go in the other direction. Our pump works just like we had before, and we get restoration of ionic balance, and we can go ahead and we can do this all again, right? So we know now there's a couple differences. One, chloride comes in immediately after sodium depolarizes. Two, calcium rushes in, activated by free calcium outside, and that plateaus our system for for an extended period of milliseconds. And then we finally repolarize and do things kind of normally. So why do we have this plateau and what's different? Well, with skeletal muscle, remember, we had an all or none principle applied to individual muscle fibers, all right? They had to be stimulated. An individual muscle fiber was stimulated and would either twitch fully or not twitch, right? But remember the whole, and we could get the excuse me here, let's go back to here. We could get a contraction and a relaxation, and we could get a building until we got to tetanus of a muscle, right? But individual fibers either contracted or not, okay? They had very short refractory periods as well, about 1 to 2 milliseconds. Cardiac muscle, we want to have a all or none principle for the whole organ. We do not want the heart muscle to contract a little and then contract more and then lock in place like we see here. The heart either contracts as a unit or it doesn't contract at all. And this is due to those gap junctions, right? Where the heart kind of beats as one. Okay, so what we want to do is we want to have a longer refractory period where we cannot cause contraction to occur on top. And so this long refractory period decreases the chance of tetanus, and this long refractory period is what we see right here in the plateau phase. Your book, the one thing that I do like about that diagram is it shows here that the absolute refractory period is this very long time frame. If we were looking at an action potential in skeletal muscle, this would go up and down very quickly, and our refractory period would be very short. We would be able to build on that refractory period and get tetanus. We don't want that. We don't want tetanus. We don't want our heart contracting, 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 and locking. We want our heart to contract fully once, have a relaxation period, and then contract fully again. So that's why we see some of those things. Now, how do we get the electrochemical signals that we need to send throughout the heart so the heart will beat kind of as one? Well, we need a conduction system. And the conduction system is the way in which we're going to send electrical signals through the heart. So the autonomic nervous system innervates the heart, but it only innervates it to modify the rate up or down. So we actually tend to start with a particular heartbeat rate that our heart normally beats at, and then the autonomic nervous system will increase or decrease that, okay? And this is really key. It does not initiate. The autonomic nervous system only modifies its rate. It does not initiate contraction of the heart. Where contraction of the heart comes is from pacemaker cells, and these are autorhythmic cardiac cells that have unstable resting potential, or we call it pacemaker potential. And these guys spontaneously depolarize at regular intervals without external innervation. Now, if I could clamp your heart off away from your body, from your autonomic nervous system, keep it in your body, but just clamp the vagus nerve and other nerves away from it, your heart would immediately go up to about 100 beats per minute, because that's what your normal pacemaker rate is. Now, we don't often have our heart at 100 beats per minute. I'm sure right now my heart, even though my heart rate's up a little bit because I'm actively lecturing, I'm probably at about 80 beats per minute, which means that my parasympathetic system is damping down on those pacemakers, telling them beat less, all right? But they would normally spontaneously depolarize around 90 to 100 beats per minute. All of these cells are nerve-like contractile cells. They're actually modified muscle cells from the cardiac system, right? And that's important, okay? This intrinsic cardiac conduction system are cardiac cells, muscle cells, that are nerve-like. They're muscle cells that have converted themselves to just act like nerve tissue. So these cardiac muscle cells make up this intrinsic conduction system, and they are going to cause the electrical impulses to go through our system here. Now, this picture here is on page 687 of your notes. And if you want, you can go ahead and write in what I did here, which is what's in the book. So you see, 1, 2, 3, 4, 5, the 5 different parts. And I put 1, 2, 3, 4, 5 up at the top here as well. And I wrote in the names. And then I just colored in some things here. Now, what I'm going to do is I'm going to go over this, but I'm going to do it using the nice color version of the book, okay? So if you want to pause and write any of that stuff down, great. Otherwise, I'm going to switch over now, and I think I can get to it just a couple pages back here. Yeah, there's the innervation part. And I think if I just scroll up a little bit more, there it is. Okay. So we're going to zoom in a little bit and get this so you can see a lot of it. And we're going to cover it now, and I'll just use my cursor and point at things. Okay. So the very first thing that happens is there's two nodes of this pacemaker cells. The first one is the sinoatrial node, which is our true pacemaker, what we think about as our pacemaker, and it generates an impulse. And this is the one that would generate a really high rate if we didn't tamp down on the heart rate. It's going to send its signal across the muscle tissue, both the right and the left atrium, and it's going to cause contraction of the atria to occur. Okay? And if you look at your lecture notes on page 9 down below, let me switch back over there just for a second. I'm going to now start kind of talking about this little bit right here. Okay? So atrial contraction is when the SA node, the pacemaker, depolarizes, an electrical wave speeds across both atria, and then atrial contraction occurs. So first the electrical impulse goes in both directions, and then shortly after, contraction starts. Right? Let's go back to our book here. And one of the places that it goes to is it goes right here to the AV node. And that AV node will now send that signal in a very coordinated way down the interventricular septum here. And it goes down what we call the AV bundle, right? And then that bundle will branch into two called bundle branches. And this electric signal will now go all the way down to the apex. And then it will turn, and as it turns, it goes up the walls of the muscle, and it will go into all of these smaller subendocardial conducting network, which we usually call Purkinje fibers. I've named that for a very long time. And we will then get contraction starting down here and going across in a wave across the heart. So what we see first is we see contraction of the atria, and then a quick pause, and then we see contraction going up of the ventricles. So I'm going to switch over to me for a minute. So if your heart is right here, the two atria will contract, the electrical signal will go down to the apex and start to go up the Purkinje fibers, and at the same time both ventricles will start contracting in almost a twist-like fashion. They kind of contract like this, and they're going to force blood up and out through the pulmonary semilunar valve and the aortic semilunar valve to the lungs and to the body respectively. Okay, so this is the aortic one, goes out to the aorta into the body, and then it's not shown here because it's underneath the pulmonary semilunar here, going to the pulmonary trunk. So we've got all these steps. So if we go back to our notes now, we have atrial contraction. As we said, the SA node depolarizes, an electrical wave spreads across both atria, then the atrial contracts. Then we have ventricular contraction, and this starts at the apex in a twist-type fashion, and it goes towards the atria, and it ejects blood through those semilunar valves. Okay. Now, I'm not going to cover the heart block stuff too much right now in class. We're going to cover that in when we do a little bit of ECG, and actually some of this information here is not entirely correct. Needs to be reworked, but I will, so I'm going to skip it for now, and I'll let your lab instructor kind of go over the heart block stuff. I can say this, that heart blocks are often damage to the AV node, or there's conduction interference from the SA to AV node. That's certainly true, and that can cause problems. We won't use, the whole conduction system won't work. But one thing I will mention quickly is arrhythmias are when our heart is beating irregularly. And this is often because the ventricles and atria beat in an uncoordinated manner, and that leads to fibrillation. And fibrillation is an irregular and fast heartbeat, and it's when those ventricles are out of phase, and we're trying to kind of fix things. And in doing so, the heart will just kind of beat really fast, trying to pump blood out, and it's not working very well. I often kind of think about this uncoordinated manner is like, you know how when you dry like towels in your dryer, and you get a bunch of towels, and they all kind of bundle up together, and they make they all go into one part of the dryer, and now the dryer is out of whack, and so you start hearing the thump, 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 and the dryer's vibrating and not working correctly. That's kind of what these arrhythmias and fibrillations are like. The atria and ventricles need to contract. Atria contracts first, then the ventricles contract, and then they relax. And keep doing that. And if they're out of sync with each other, blood is not going in and out correctly, things are getting not working right, and the heart is inefficient. Just like your now dryer is being very inefficient in not doing its job. So that is kind of what I think about when I think of those things there. All right, now what we're going to talk about next is ECG, electrocardiograph, often referred to as EKG still. That's the German spelling of cardia instead of cardiography with a C. So I will refer to it as ECGs, but you'll hear it as EKG probably out in the world. That's fine. What an ECG is a way for us to watch things going through this transmissions, this intrinsic conduction system, going from the SA node to the AV node, going down the bundle branches and out the Purkinje fibers. And there are three main waves that we see here, okay? There's actually four waves. There's two depolarizations and two repolarization events. However, one of them is hidden, and we'll talk about that in a minute. All right, so the first thing here is we want to look at the ECG. And what you're noticing is you see three waves. You see a P wave, you see a QRS wave or complex, and then you see a T wave. Okay. Now, what do all those mean, or what are all those things happening? Well, let's think about what we just talked about, and let's think about what each of these do. So the P wave is atrial depolarization. This is when the spread of the impulse depolarizes from the SA node through to both left and right atria and to the AV node. That's what's written in there, to the AV node. Okay, this takes place about 1/10 of a second after P wave initiation is the contraction. So here's where the initiation of the P wave is. Contraction occurs a little bit later. It occurs about 1/10 of a second later, right usually here at the peak of the P wave. And that's just because it takes time for the electrical impulse to go into the cardiac muscle to cause actin and myosin to come together and start to shorten. So the P wave talks about the impulse going over, and we know that shortly after that, the atrial contraction occurs. Okay? Then we have the QRS complex. Okay, what's that? The QRF complex is where we have the AV node now sending the message down to through the apex, through all the Purkinje fibers, and we depolarize both ventricles. So the QRS wave is ventricle depolarization. All right. So this takes a little less than 1/10 of a second. Now, at the same time, the atria have already contracted. They are repolarizing. Okay? So atria are repolarizing and are not seen because it is happening behind the QRS complex. So this thing that says masking, it means that the repolarization is actually occurring. The repolarization is occurring behind the QRS. It's not part of the QRS. It is literally hidden behind it here. We can't see it. That's the fourth wave. We have one wave here, depolarization atria. Repolarization atria is hidden from view. The QRS is stronger and goes over top of it. Okay? So that's interesting. Now our third wave is the T wave. And the T wave is when the ventricles repolarize. Now that we can see because the atria have already repolarized. Nothing else electrically is happening. We need to bring all the sodium and potassium back to their right place. We have to get the calcium back in the SR. We do all that stuff for the ventricles. This is the T wave here. So again, the repolarization starts and the relaxation occurs slightly after. There are a couple intervals and segments of this that we measure to see if something's going wrong. And you'll look at some of these in lab when you do the ECG lab. But I will cover a couple right now, and then we'll stop here. And I think maybe what we'll do is we'll take this will be where we end this lecture, and we'll pick up and do one more going over the cardiac cycle and some of the electrical stimulation of the heart. So let's look at these three really quickly. So the PR or PQ interval is from the beginning of atrial excitation, okay, to the beginning of ventricle excitation. All right, so this is the time required for the impulse to travel all the way through the atria, depolarize and repolarize, and the AV node. Okay, so the PR, PQ or PR, it really goes to Q, so it's better to say Q. And so it starts here at the P, beginning of the P, and it ends right here at the end of, or at the beginning of the Q. And so we can measure that, and that tells us how long the depolarization throughout the entire atria to the very beginning of the ventricle depolarization occurred. Okay, so that's the PQ interval. The second one is the ST segment. And this is beginning at the end of the S wave, beginning to the T wave. Right? So at the end of the S, the end of the S wave to the beginning of the T wave. And so this is at the end of the spread of the impulse through the ventricles and the repolarization. So this is when, this is the important part, because it's at kind of the end of depolarization and the beginning of repolarization. This is when the ventricle contracts and ejects blood. And so the ST segment is approximately from about here. It's not very, I don't like how this is really well done, but at the end of the S and to the beginning of the T, that little gap there, that's when ventricle contraction is occurring. Okay. All right, now the last one is the QT interval. So the beginning of the Q till the very end of the T wave, right? This thing is the entire ventricular contraction from the beginning of contraction through repolarization. So the QT interval is the entire process of ventricles contracting and relaxing. And so that's a useful one to look at too, because your ventricles are so important to pump blood to your lungs and your heart. We want to make sure that that is correct interval. I'm going to come back to me. We're done now with this, and I'm going to just say a little thing and we'll come back. So an example of this where it's important is my background is in human genetics, and I used to work at the Human Genome Project for the University of Michigan, where I went to school. And one of the things I got to do was we would get samples from patients, and we would try to find genes that cause genetic diseases. And so one of the diseases I worked on many, many, many years ago now was this thing called long QT syndrome. And so in long QT syndrome, literally patients who have this disorder have a very long QT interval. And so their QT interval is so long, and it takes so long for the heart to kind of contract and eject blood out of the ventricle that there's something wrong. And so they don't have a very efficient heart. It's very slow. And if you can imagine trying to do exercise, go running, play sports, and you have this really slow contracting, long interval for your ventricles, it wouldn't work very well. And so what we were doing is we would go and we would get samples from patients who had this disease, and we would get their family members, and we would try to figure out the genetics of that disease. And so I worked on that for a while. And so it was interesting because I didn't know what long QT syndrome was way back then. Until later when I started to study the ECG and say, oh, so that is what I was doing my research on when I was a lowly student many years ago. I was looking at individuals who had an exceptionally long QT interval. So kind of cool. All right, well, we're going to stop there, and we'll pick up in our last lecture some of the cardiac cycle and some of the other things about the heart. Thanks.